Hello friends, I am Dr. Suhas Talmale and today we are going to see about transgenic animals. The natural genetic makeup of an animal is modified, is changed, is manipulated by transferring one or more genes in it in such a way so that its milk production, if it's cattle, milk producing animal, its milk production will increase or the quality of milk will get improved. Example, its protein content or vitamin content might be increased. Wool content of the sheep or many other like meat content of the poultry birds. Such things can be performed by transferring a new gene, a foreign gene in that individual. Of course, it's a part of recombinant DNA technology also. Because in that, either you will take a vector or it can be a physical gene transfer method, which is also called direct gene transfer method. Any of the method, vector or without vector or vectorless method, direct method, or it can be any chemical method of gene transfer by which the quality of the product or the quantity of the product can be improved or increased. If the gene transfer is transient, with every generation the activity of the gene will decrease, and after a few generations it is well possible that it might lose that transgene. But in case of permanent, we are getting a permanent transformation, a permanently transformed, a permanently improved type of genetic makeup. And here, generally this is the aim for most of the time, because what do we want is a permanent change. What are the objectives? First, to study the functions of transferred genes. Many a times, many genes are not well known. So we want to study their functions, and for that we can form any transgenic animal and can see whether what is the effect of this gene. Then to obtain protein from transgene. Of course, for that also we can go for that. We can transfer any gene in any particular animal, and if in its urine or if in its milk that particular protein is formed, is secreted, then we are actually producing that gene, that protein in that animal. This can be called bioreactors. Genetic modification to improve quality or quantity of milk, meat, wool, etc. Now, this thing we have just covered. To obtain protein product in milk or meat. Then gene therapy. Many times, many people are suffering from some genetic disease, diabetes mellitus, in which the person is lacking or having a defective copy of the insulin gene. But if we can, by some or the other effort of gene transfer, if we are able to transfer the gene of insulin synthesis in it, definitely that will improve it, and such therapy is called gene therapy. The next objective is specific transgenic animals can be synthesized or specific cell lines can be synthesized to fulfill specialized experimental needs. Suppose we want any particular type of animal which is overproducing triglycerides, because I want to learn obesity or any defect in fat metabolism, and suppose I want to form a rat for experiment which is overproducing the triglycerides, then definitely the gene for triglyceride synthesis, if I will transfer an extra copy of the gene for triglyceride synthesis, definitely the triglyceride synthesis may increase, and thus I will get a diseased rat. If I will get one male and one female of this type, I will mate them, and of course I will keep on getting next generation which are having high triglyceride synthesis than the normal. diseased rats. So in this way, what we have designed a particular type of animals which are going to fulfill our specialized experimental needs. Similarly, and such mice or rat or any animal are called knockout. There are four main methods, the four most successful methods. Keep in mind, there are many methods by which transgenic animal can be synthesized, but the four most successful methods we are going to see. The very first in that is retroviral vector method. In that, we are going to form a retroviral vector. You can see this is the SV40 plasmid vector, and we are going to transfer the gene of interest in this plasmid using recombinant DNA technology. Means actually recombinant DNA technology is a small part of transgenic animal cloning. These are the big science, and RDT is actually a very small part of it. Look at it. We have plasmids of this type, in which we have ampicillin resistance gene, which will be used for selection as a marker and will be useful for selection in case of E. coli or prokaryotes transformation. PBR322-ORI, which is the origin of replication in prokaryotes. SP40-ORI. SP40 is a virus which can easily infect humans, and from this virus its ori is taken. Then with the help of it will divide in, or it will replicate in humans or mammalian cells. There is SV40 promoter also, promoter on which binds RNA polymerase, and transcription of the gene will take place, because immediately after this promoter there is a gene of interest. Here actually we have transferred the gene, followed by poly A site, because the mRNA which will be formed will be very stable if we will have poly A site after it. Now this is what? This is a recombinant vector which is having PBR322-ORI as well as SV40-ORI, means it can replicate in prokaryotes as well as eukaryotes. We can call it shuttle. Nowadays, MAC or HAC, that is mammalian artificial chromosome or human artificial chromosome, are also used instead of this retroviral vector, because after all it is having retroviral origin. But here in the mammalian artificial chromosome or human artificial chromosome, even YAC, that is yeast artificial chromosome, can also be used. In them we have centromeric region as well as telomeric regions, and that's why they are more stable in eukaryotes. So such vectors are also available, and they are nowadays alternate strategy for using retroviral vectors. Suppose we have a cow which is a donor female, we will provide certain hormones to it, like even we can give the FSH and LH injections also. Because of that, she will donate more amount of ovules. After that, that female is mated with a male. As mating will take place, definitely after a few days she will develop embryo. These embryos at 4 to 8 cell stage are removed from the uterus of the female by flushing the uterus with a particular solution, and these embryos are incubated with this retroviral vector. This retroviral vector many times show transfection, and that gene is transferred. Now this embryo or such embryos are implanted back into the same female either, but if her uterus is disturbed during flushing, we can take surrogate mothers also, which might be the close relative of the same female. And this female, which is a surrogate female, is also mated with a vasectomized male, so that her uterus will become ready for the implantation of the embryo. When we talk about vasectomized male, means those males in which vas deferens is blocked. The male is unable to transfer his sperms to the female, and that's why the such female are called pseudo pregnant. In case if the same female is ready, her uterus is not disturbed during flushing, we can transfer it in the same female. In case if we have many embryos, because we have super ovulated this female by giving FSH and LH injections. In case this female has given more ovules and more embryos are there, then a few are transferred in the same female, a few are transferred in the surrogate female. Now, after few months, it will deliver the child, and it might be non-transgenic, means though we have tried it, but the gene is not transferred, or it might be a transgenic founder. When we talk about founder, means the chances of this are less, and that's why such a genetic makeup in which the chances of getting such a genetic makeup is less, we call it transgenic founder, while the chances of getting non-transgenic are more. Now, in the next generation, suppose we have a male transgenic founder, a female, if we will get one male and one female, that is sufficient, because we will mate them and we will bring out the next generation. Now here, the only problem is retrovirus can become virulent also, because this retroviral vector, in that the retroviral things are mutated, and that's why generally it is not possible to become virulent. It's an invirulent type of viral vector. But sometimes any mutation in it can make it virulent and it can become dangerous. This is a major problem, especially when we are using this retrovirus in animal like cow, of which milk is the most important product and which is we are going to consume. And this is how retroviral vector method is there, by which the transgenic animal can be synthesized. Microinjection is another method. Now here again the super ovulated female is there. Super ovulated, which will be given either HMG, human menopausal gonadotropin, or FSH or LH injection, so that she will deliver more ova. Instead of 5, she might give 35, means at least 5 to 7 times. Then that female is mated with a male. Now a female, suppose we will take an example of sheep. If we have super ovulated female sheep and we will allow it to mate with male, then definitely she will have much more fertilized eggs in her uterus. But in case if mating is not possible, we can go for in vitro fertilization also. Means we can remove the sperms from male and we can remove the ovules, because the female is super ovulated, so she will give more ovules, those ovules from female, and we can go for in vitro fertilization also. That is also possible, but in both the cases fertilized eggs are possible. Now here we won't allow this fertilized egg to go in 2-celled or 3-celled embryo form. Before that we remove it. Means immediately one day after the mating we will remove the fertilized egg, and you can see still, because within 24 hours the male nucleus, pronucleus, and female pronucleus are not still in the fused form. Once they will fuse, we will call it zygote. But here still they are not fused. Means the male pronucleus, you can observe, it's comparatively bigger as compared to the female pronucleus. And now with the help of a microinjection in which we have a holding pipette. So suppose in a petri plate or in a beaker we are observing such fertilized eggs under microscope. We have a holding pipette through which we will hold one by one the fertilized eggs, and through other hand we have micro injecting pipette that is having a very thin and very fine needle, and that through that we will transfer the gene. We will pick up the copies of genes and we will transfer, and this fertilized egg in which gene is transferred, that is transgenic fertilized egg, are implanted back, where either in the same female, but the female was super ovulated, so we might be having many more zygotes, In that case, we might need to go for any surrogate females also. Either surrogate, which are again made pseudo-pregnant by mating with vasectomized male, and then her uterus will be ready and in it is implanted. After the delivery, the next generation is checked for transgenic, but again most of them are non-transgenic and only few transgenic founder because again the population is quite less. That is found. Now how it is checked? A small piece of the tail is cut, and from that DNA is isolated, PCR is performed, and after matching the DNA fingerprinting, we will came to know that these are the transgenic founders. There are certain disadvantages also. Trained people are required. No one, though it looks quite simple, but it's not as simple, and that's why trained, who have taken proper training, only through them this method can be performed. Success, the chances of success are too less. Out of 1,000 events, only 30 to 40 are found to be successful. And the major problem is random site. This transgene, though we are transferring directly into the nucleus, it can go in random site. It might go to a silencer region where the gene will not be expressed. So it is well possible that simply transferring the gene is not sufficient many a times. So this is about the microinjection, especially male pronucleus, because it is comparatively bigger in size. The third and widely used method is liposome mediated, also called liposuction. First of all, what is liposome? Liposome is a small vesicle made up of lipid bilayer, just like plasma membrane. So, suppose I have to draw any liposome, I will draw it something like this. We are well familiar, these are the lipid molecules which we are showing, and this is the lipid bilayer. And if in a similar way I will complete this complete circle, this circle of lipid bilayer is called liposome, and it is represented like this. This is the lipid bilayer, exactly just like plasma membrane, and inside it there is a transgene, the gene which we want to transfer in this cell. So this is the plasma membrane or liposome, which is having its membrane just like plasma membrane, and this is the cell which we want to transfer. This is the nucleus of the cell. If in presence of this cell we'll keep this liposome, and suppose chemicals like fusogen, example polyethylene glycol is a fusogen in the presence of which the chances of fusion will increase. In this case, it is well possible that this liposome will go close to the cell, and this outer to outer membrane will fuse and inner to inner, because plasma membrane maintain fluidity, and that's why when this lipid bilayer will come close, it will fuse and transferring this transgene inside the cell. This is how transfection can take place. Another physical method, but it also uses certain chemical like polyethylene glycol, and thus the gene is transferred. But don't forget that we don't want to transfer the gene in the cytoplasm. We want to transfer it where? In the nucleus. But here it is transferred in the cytoplasm. That's why a new technique, that is liposome within liposome, is used. In that we have one liposome, and inside it there is another liposome, and here the aqueous fluid is filled, and here inside this liposome, the smaller liposome, we have transgene. Now when the outer layer of the liposome will fuse, it will give a chance to the inner liposome to enter this cell, and now inner liposome with transgene will be present in the cytoplasm. Now suppose this liposome will fuse with the nucleus, definitely the nuclear membrane will give it away and the transgene will be transferred inside the nucleus. And for that, what do we need to do? We need to introduce certain ligands in this inner liposome for which receptors are present in this nuclear membrane. Because of that, this ligand and this receptor, as they are perfectly complementary to each other, they will come and bind, they will go close to each other, because this inner liposome might fuse with mitochondria also or ER also. To make a directional approach towards nucleus, we need to introduce certain ligands so that exactly the receptor of those ligands will be present in the nucleus and they will go and fuse, and the chances of this liposome, inner liposome, to fuse with nucleus will be more. This is how gene can be transferred. This method is comparatively easy. It will take place in 3 stages. First, adhesion. Second, fusion. And third, release of the DNA inside. The fourth and most important method, the latest method of forming transgenic animal is this one, in which we are going to use embryonic stem cells. Embryonic stem cells which have the ability to form a complete individual, because they are embryonic, they are still not differentiated. Now, this is the donor blastocyst. Means what? There is a male and a female. Of course, mating will take place. After mating, the female will become pregnant, and from her blastocyst, we are going to remove certain cells. These cells in the blastocyst are embryonic stem cells, because stem cells which have the ability to form a complete individual, and that's why they are embryonic stem cells. And now, with embryonic stem cells, we are going to incubate the transgene. This is the transgene. Many copies of this transgene with embryonic stem cells, they are kept with each other, and transfection takes place, in which this gene is transferred in the embryonic stem cells, and some of them get transformed while most of them remain non-transformed. The transformed are selected on the basis of marker, because along with this transgene we also have marker, and on that basis, from the marker we will select the transformed, and you can see only transformed are there, and these transformed cells are grown like a cell line, and they are microinjected in the blastocyst. Which blastocyst? Other blastocyst. Either in the same female or its relative female, close relative female, which is also made pregnant. Transformed embryonic stem cells are microinjected back. And this blastocyst, and such many blastocysts can be formed, these blastocysts are implanted in mice. Mice or any other animal. We have taken an example of mice. It can be sheep also, it can be cattle also, and in that, when it is re-implanted, that female will give birth to the individual. Most of them are again non-transgenic, means though the gene is present, it is not expressed. But a few might be the founder, transgenic founder, and these are selected, and this is how this method can be used for forming transgenic animal. Again, in any of these 4 methods, if we have a male and a female, mating can be done and next generation can be formed. In every generation, we have to check whether it's a stable transformation or a transient one. If it's stable, we have successfully created a transgenic animal. This is all about transgenic animal. Thank you.